Welcome back to the deep dive. Today we're digging into the sources you sent over, and we're tackling a very cold topic. We're talking deep freeze management of some of the most fundamental materials in biotech. You gave us material on a really fascinating shift in how manufacturers handle microbial cell banks, the MCBs and WCBs. The old way of thinking, and you still see this in some of the regulatory language, is that these banks are just static, like a file you archive in a freezer and it's just unchanging.
And that static assumption is exactly where the risk is.
Yeah.
What the leading firms now get, and what your sources really highlight, is that a cell bank is a dynamic biological system. It's always evolving.
Evolving how, though? I mean, these are cells kept at -80, even minus 130 degrees Celsius.
Well, even in that deep freeze, the population is under stress. You've got cell age, what we call passage history. You have the stress from the actual cryopreservation process, and even tiny changes in storage conditions. All that adds up. It means the bank's performance, how it grows, its stability, even the impurities it might produce can just drift over time.
So if you're managing them passively, just assuming the data from 5 years ago is still good, you're basically introducing chaos downstream. What does that actually look like on the production floor?
It looks like batch failure. It can look like inconsistent fermentation kinetics. So one batch hits its target in 120 hours.
And the next one takes 140, right? It just throws the whole schedule off.
Exactly, the whole schedule. And even more critically, it can mean altered impurity profiles or changes in the critical quality attributes, the CQAs of the drug itself.
Okay, let's quickly define CQA for anyone who doesn't live and breathe quality by design. What are we talking about here?
Right. So critical quality attributes are the specific features that really define the drug's safety and effectiveness. Things like purity, its potency, making sure the molecular structure is perfect. If the cell bank shifts, you risk changing those CQAs, and that means incredibly expensive investigations or just throwing the whole batch out.
The stakes couldn't be clearer. So our mission for this deep dive is to really unpack the framework that solves this, applying pure manufacturing logic, quality by design, FMEA, statistical process control, directly to these living materials.
Precisely. It's about moving cell banking from a pure microbiology task to a lifecycle managed, qualified raw material, something essential for a robust process. The foundation for this whole shift, as the sources lay out, is using a systematic risk matrix. It's straight out of FME thinking. It lets you map every single parameter, operational and design, to all the plausible failure modes.
So it's not just about what happens in the freezer anymore, it's about what happens on the factory floor. The goal isn't just, is the cell alive, but how does that risk travel into real metrics like yield, scheduling, and of course, regulatory defensibility? This is about protecting a multi-million-dollar process. And in that framework, the material you sent highlights two really critical risk parameters that honestly are often missed outside of these specialized teams. First one is tracking cumulative cell age. It's usually measured in population doublings or CPD.
Yeah, this is a huge one, so often underestimated. Biologists know cells age, but in manufacturing, you have to quantify it. The rationale is pretty simple. The higher the CPD, the more chances you have for a random mutation, or for the cell to just, lose the plasmid, the genetic instructions for making the drug. Or you start selecting for these weird, slow-growing subpopulations that have adapted to stress.
So if a manufacturer is using a cell bank that's getting a little old, getting close to its CPD limit, what's the control? What do they do?
The control has to be incredibly rigorous. First, you define a maximum allowable CPD based on studies, and then you have to constantly trend your process performance, your titer, your CQAs, against that cell age. The real power here is in the regulatory conversation. When an inspector asks, is your cell line still good after 5 years? You don't just say, we think so. No. You show them 5 years of data. You prove that the line is still performing robustly at, say, 70% of its defined maximum age. It totally changes the conversation.
It moves it from a defensive posture to a proactive proof. Okay, so the second critical point you flagged was the regeneration pathway. The SOPs must explicitly forbid something called back generation. Why is that one so bad?
Back generation is a huge, huge risk. It's where, instead of going back to the original pristine master cell bank, you try to make a new working cell bank from maybe a production seed culture, or even worse, from the main fermentation tank.
So it's a shortcut.
It's an unverified genetic shortcut, exactly. It introduces a massive, uncharacterized amount of age, and who knows what kind of contaminants might have crept in during production. If you can't trace that vial you use today directly back to the original MCB lot, that is a major red flag for any regulator, a huge non-compliance issue. The risk is you've now got a subpopulation that is genetically different from what you started with.
It's a break in traceability. It undermines the whole quality system. Okay, let's shift gears a bit. Let's talk about the physical controls, the engineering that goes into keeping these cells fit during the whole freezing and storage process.
This is the moment of truth. Freezing creates massive stress, and how you do it really determines the bank's future. We need to talk about cryoprotected formulation and the freezing rate. If that formulation is off, or if you freeze it too fast or too slow, you get ice crystal formation. It physically damages the cell membranes. And the sources are clear, this doesn't always kill the cells outright, it just leaves them really, really weak.
So what's the consequence of that in the factory? A bunch of weakened cells.
It leads directly to poor post-thaw recovery. We track that with the CFU count, the colony-forming units. Basically, it's how many viable cells can actually wake up and grow after being frozen. If that count is low or variable, your seed growth is going to be all over the place. And that means inconsistent inoculation of your main tank. You've just started a multi-million-dollar batch on a totally unpredictable foundation.
And it's not just about being alive, right? We have to talk about identity and genetic integrity. The cells have to remember what they're supposed to do. What's the biggest risk there?
The risk is construct loss.
Yeah.
Or rearrangement. That plasmid, the little circle of DNA with the blueprint for the protein, is exochromosomal. Cells could just drop it. Or parts of it get rearranged and suddenly you're making a protein that doesn't work. And this isn't just about yield, about lowering your titer. It can actually change the product's CQAs. You might create a truncated protein that's inactive, for example. So the controls have to be tight things like sequencing or genetic fingerprinting, and specific plasmid retention assays at defined passages to prove the cells are still holding on to the payload.
Okay, and finally, there's the physical security piece, which sounds basic, but a failure is catastrophic. Storage configuration and environment.
Yeah, the standard controls are what you'd expect. Validated storage temperatures, continuous monitoring with alarms, making sure you stay way below that critical glass transition point, usually minus 130°C or colder for long-term storage.
But the sources really hammer this home, this idea of mirrored storage across dual freezers.
Oh, it's absolutely essential. It's pure risk mitigation. It's about avoiding a single point of failure. If one freezer dies, power loss, vacuum leak, whatever, you lose half your bank. But the other half, in a separate unit, maybe even on a separate power circuit, is still viable. And physical segregation is just as critical. You have to make it impossible for an operator to accidentally grab a vial from the wrong product line. It's a simple human error, but one with massive consequences.
All right, so we've covered the design rules like CPD, the physical controls for freezing. Now we get to segment 3. This is the real test of that engineering rigor, release testing. Moving beyond just counting cells, confirming they are functionally fit for purpose.
Right. It's about function, not just survival. And the sources show that the most predictive tests measure dynamic growth kinetics. There are 2 core metrics that tell you exactly how those cells are going to behave in the bioreactor. The first is post-thaw lag time. We call it lambda. You can think of it as the cell line's wake-up time. How long does it take for the cells to adapt to the liquid media and really start growing exponentially?
And the second one is specific growth rate, or MU.
Yep, mu is just how fast the cells grow once they've woken up and are in that exponential phase. Any shifts in lambda or mu are critical because they predict everything about the large-scale batch days later. They tell you when to feed, when to aerate, when to harvest. If lambda or mu are off, your whole batch timing is off.
Okay, let's unpack the decision logic here, because this is where the system really shows its value. The sources have this great example, a recovery kinetics matrix for an E. coli bank. So if lambda and mu are both fine, it's a routine release. Easy. But what if? What if the bank has a slightly high lambda? The cells are a bit slow to wake up, maybe an hour longer than usual, but their mu, their growth rate, is totally normal once they get going.
That's a great scenario. It suggests some mild stress, maybe an age effect, but it's not a catastrophic failure.
Yeah.
The matrix says you don't reject it. Instead, it becomes a conditional release. You use the material, but with mandatory enhanced monitoring in the early stages, and you launch an investigation to figure out why. You check the storage history, for instance.
So you get to use expensive material, but you're acknowledging and managing a minor risk.
Exactly. That is the difference between an engineered system and a simple pass-fail checkbox. You can quantify and manage that risk.
Okay, now for the obvious failure. What if the bank shows a high lambda A&D, a low mu?
That's the kill signal. That combination means significant functional impairment. The cells are slow to wake up and they're slow to grow. That bank is basically guaranteed to cause failed seeds, off-spec batches, and total chaos for scheduling. The disposition is non-negotiable. Reject the bank lot, the material is unfit, and a root cause investigation starts immediately. It takes human guesswork out of the equation.
And you also mentioned that high variability between vials is a big red flag, even if the averages look okay. Why is that?
Because variability means the population is heterogeneous. It's not uniform. It might be splitting into high fitness and low fitness groups. If you pull two vials, they could behave completely differently. And that means you can't rely on any of them to perform to spec. It suggests major damage or instability, so you have to put it on hold and investigate.
This all brings us to the long-term view. I mean, these banks are supposed to last for decades in a freezer. How do you possibly manage that performance trend over such a long time? The answer, of course, is statistical process control, or SPC.
SPC provides that mathematical rigor you need for long-term confidence. You pull retained vials every year or so, you test them, specifically tracking the log euro CFU values, and you plot them on control charts. This lets the manufacturing team spot degradation years before it would ever cause a crisis on the floor. And the sources lay out some really clear SPC rules for this. Let's look at two of them because they define two very different types of failure.
Okay, first one. Rule 1, the catastrophic failure. This is when any single data point falls below the lower control limit on the chart.
Right, that's a high-severity, low-frequency event. It points to a sudden, major failure. Maybe a freezer had a brief temperature excursion, a power dip that compromised the whole unit. This triggers an immediate full-scale investigation, probably a regulatory notification, and you might have to stop using that entire bank lot right away.
And then there's the much sneakier one. Rule 2, chronic degradation. This is a trend. Specifically, seeing 7 or more consecutive points all fall below the center line.
This is the predictive rule. The cells aren't dead, they haven't hit the lower limit yet, but the statistics are screaming at you. The trend is clear. Viability is slowly but consistently declining. This triggers a plan for proactive bank replacement. You don't wait for it to fail. You retire it based on the data showing it's in a state of chronic drift.
So this whole framework, the FMEA risk assessment, the dynamic release tests, the long-term SPC trending, it really does close the gap between, old school microbiology and modern manufacturing science. It builds resilience right into the process. It ensures that biological integrity is actively managed, not just passively assumed.
It makes sure that your biological starting material, your foundation, stays aligned with your process intent for years and years. The whole mindset shifts from relying on history to actively monitoring the fitness of the material and predicting its end of life.
So what does this all mean for you? The big insight here is the shift from treating something as static inventory to seeing it as a dynamic system, a system that needs constant monitoring and risk-based plans for replacement. And since even the most stable, deeply frozen biological material needs this level of engineering to maintain quality, it begs the question, what other critical materials or specialized components or even long-term assets in your professional life are you currently treating as unchanging inventory, when in reality they are dynamic systems slowly trending toward a predictable chronic failure? That's the mindset shift to think about from this deep dive. Thanks for diving deep with us. We'll catch you next time.